biologists do. People are paid to do what you're going to do. So I hope it'll make you feel really good about your skills when we're finished. So yeah.
Are we actually starting them today, or we want to introduce them?
We're starting them today. Yeah. Yeah. So we've got a couple of handouts to help us in this process. So each of you guys have grabbed each of the handouts. The white one is a little kind of it's kind of like my notes that I made for the procedure. So a little flow chart. I'm a real visual learner, so when I'm doing procedures, I like to make little cartoons like this. And then the second one are the dichotomous keys. These are the keys you'll use to use your test results to identify your 2 unknown bacteria down to the species of them. Okay? So what I'm going to do is, unfortunately, I'm going to give kind of a lengthy introduction, and the reason is once we start, I can't help you anymore. So one of the important rules here, so our rules here is that this exercise, it's totally open book, open notes, open computer, open notes. You can use your computer, you can use the internet, any kind of resource, except you cannot use another Homo sapien. Okay, so other human beings, you cannot ask for technical help, okay? But you can use any other resource that you want. All right, so technical help means you can't have someone help you figure out how to do a gram stain. You can't have somebody look at your bacteria and tell you, are they gram negative, are they gram positive, what's the cell shape, what's the cell size. You can't ask somebody, how do I run this test? Which media do I use? You can't ask somebody, it's like, will you look at my MR tube and tell me if this is positive or negative? Okay, so you can talk. You can talk. In fact, one semester was so hilarious. I mean, it was hilarious. I gave this introduction, I said, you can't help one another, blah, blah, blah. And this was a group, like you guys, they got along well, they like to talk with one another. So we started the exercise, it was dead silent. I was like, what's wrong? And so people thought they couldn't talk. And it's like, no, you can talk. You just can't ask for technical help. And this is going to be really important, you guys, that you help one another, because don't put your colleague in a hard position by asking them for help. Remember, I mean, your colleagues want to help you, right? But they aren't supposed to give you technical help. So please don't put your colleagues in a tough position and ask them for help. So the way it works is if I see that information, technical information is being exchanged, points will be deducted. Okay, so you don't want to do that right now. You want all your points. You deserve your points. All right, so that's just important for us to remember. The way this procedure is going to work is that each of you will get a matched pair of unknowns. And what I mean by matched pair, each of the broth tubes will have the same number. I'll just make this up, you guys. Let's say 401, 401, and so we have the same number, but one will be A and one will be B. Now, each of you in these pairs, one of these tubes will be your gram negative and one will be gram positive, but you don't know it, right? Very important for you guys to know that the gram negatives are different number to number. So some folks, their A will be their gram negative. Other folks, the B will be their gram negative. And I just tell you that just in case you hear maybe your partner mumbling to themselves, aha, A's gram negative. And you've just discovered that your B is a gram negative. I don't want you to panic thinking you made a mistake or your partner made a mistake. They're all mixed up, right? Okay. So why don't we right now, why don't we go ahead and just kind of do these steps as we're talking about them. So if you guys will pull out your test tube racks, and if you don't have enough, each person, it'd probably give each person a test tube rack. Just let us know, we'll get you one. And I'm going to come around and let you choose your unknowns right off the bat. Okay, so we'll just, you're going to have hands on while we're doing this. And the other thing, you guys, we might as well do right now, each person is going to need 4 TSA plates. So why don't we take just a little bit of time to get our test tube racks out, and also for each of you to come up and get 4 TSA plates, okay? And then we'll we'll continue our discussion of what all we're going to do today.
I'm waiting in line to get a bear vaccine.
And what I'll also do, folks, is I'll place some of the plates over here on the side just so you can spread out a little bit.
Alright. Can I get my can, please?
Oh, yeah.
Thank you.
Wait, we don't need them because we're doing augers, huh?
I think we're doing tubes as well.
Oh,
Yeah. It's not sure. Well, let's give that we have our stuff, all of them out. Yeah.
I have the leaf.
Okay. Oh, sorry.
Did anybody grab the plate?
We need 4, right?
4 each.
Thank you very much. Yeah, you get it.
Are these used?
10 points.
What? No water, what?
Make sure there's like not too much water.
Not too many water?
Yeah, that's the ones over there. Okay.
Thank you very much.
I should have told you all. You guys, please listen up on this one. This is really important. You guys, please listen up. This is important. If whenever you're coming up and getting plates, you accidentally like open the lid like that, please don't put it back. Don't use it and don't put it back. Just come over here, grab one of the and put an X on the bottom and just put it here on the desk. And the reason is there have been studies that have shown that you save time and money in microbiology. If you have any doubt at all, if something has been contaminated, don't use it. And many of you are so considerate, you don't want to waste things, right? But you will save us money, you'll save yourselves time, that if you think anything, there's the slightest chance something's contaminated, don't use it. So if you think it's contaminated a plate, like I said, just bring it over here, put an X on it. We have uses for plates that might not be perfectly sterile, okay? But please don't use it for yourself. And most important, you guys, don't put it back. Because the presumption is all these plates are sterile. And you don't want somebody else to get messed up, right? Because of a contaminated plate. Okay. Okay, each person, you guys, needs 4 TSA plates. Okay, and I'm just getting our unknowns ready here, because what I'm next going to do is I'm going to bring the unknowns around for you, so that each of you can choose your unknown. So hopefully this will make a little bit more sense as we're discussing it. So the next thing I'm going to do, this is more real world for us, I'm going to bring the unknowns around, and let me describe how they're arranged. So the matched pairs are going to be side by side. So again, what you're going to do, I'll bring the racks around, and you get to choose whichever matched pairs you want. But the important thing is when you choose them, when you choose your pairs, make sure you read the numbers, and make sure the numbers match and that one tube's A and the other tube's B. Now, when you get your tubes, the first thing you want to do is start filling in your unknown report. Okay? And the unknown report is the last page of chapter 22 in the lab manual. Okay, so your lab manual is going to be your friend. So if you look at the last chapter, which is the unknown chapter, starts, and this version, it starts on page 182. If you go to the last page, you'll see that there's an unknown report sheet. Now, I think in your lab manuals you have 2 copies. The reason there's 2 copies is many people like to use one as kind of their rough working copy, and then when they're, just before they're ready to turn it in, they like to use the second copy. They like to rework the notes, make them neater, and so you have a second copy that you can turn in as the official copy. Now, some of you are very neat just right off the bat, and you might just turn in your working copy, and that's fine, okay, but just so you have that option. Now, what I'm going to recommend you guys is if you take that report sheet and fold it lengthwise, okay, and then you open it up. And the reason that at least helps me is you'll see over on the left-hand side up at the top it says gram-negative organism. So the left-hand column will be for your observations on your gram-negative organism. On the right, you see it says gram-positive organism. So the right side is for you to document all your observations on your gram-positive organism. You're going to want to put your unknown number up at the top. Now, today you don't know who's gram-negative or gram-positive, right? You don't know if A is gram-negative or gram-positive or B is gram-negative or gram-positive. So all you're going to do right now is just put your unknown number from your tubes on the report sheet. Okay, so let me pass those out right now just so we can get going on that. Okay. And what you could start to do is on your plates, you can start labeling them. Okay, so you could put your name, the date, the lab section, and you can draw your 4 quadrants. Because today you're going to be streaking your plates using aseptic technique to get isolated colonies. Okay? There you go. There you go. Go. Now, when you guys get your tubes, make sure you gently shake them. You want to make sure the broth is turbid. You want to make sure the bacteria grew. Yeah, and you know what you can do? You see in the kill area there, you can just grab one of those. Okay?
David, can you grab me one, please?
Darker color.
Oh, okay. Thank you. Never mind.
Give me a darker one, Cheryl. It's getting too light. A dark pen. And a purple.
Oh, you don't need a rug. So we can have, we just can.
Yeah, you can't talk, you just can't ask for technical.
Okay. A darker green? What is this? Where's your keys?
Stupid.